I'm taking it a bit more practical right now. We're going to talk about the world's first neuroprotective trial in prodromal Parkinson's disease. So how are we going to approach this? I'm going to give you a bit of a background about the rationale of the trial. Then I'm going to talk about the trial design. I'm going to update you where we are on the trial at the moment. And then I'm going to let the participants of the trial, many of them are here today, give some feedback about how it is to take part in something like this. So I think the first and very important point to pause on is what do we mean when we say prodromal Parkinson's? And I think Livia did a good job at introducing this topic already. But essentially, when you're diagnosed with Parkinson's disease, it's a clinical diagnosis. And this is based on a certain combination of motor features. But at that point, you've already lost a significant number of your dopaminergic cells in the substantia nigra in your brainstem. So we know that years preceding the point of diagnosis, the disease process was already happening. And in Parkinson's, we call that the prodrome. It's about 10 or 20 years before the point of diagnosis. And during this time, you accumulate certain non-motor features. The earliest one is loss of sense of smell, then REM sleep behavior disorder, autonomic dysfunction like constipation or dizziness on standing up, and maybe some subtle mood changes, cognitive changes, and fine motor features. During that time period, your cells start accumulating this toxic protein that's misfolded called alpha-synuclein, and there are certain disease processes that propagate and worsen the situation. And these we know from animal and human models, and they are lysosomal dysfunction, mitochondrial dysfunction, oxidative stress, and also neuroinflammation. So in this trial, we are narrowing in on one specific disease process called neuroinflammation, and we are focusing on it in the prodrome of Parkinson's disease, so in someone who do not yet meet the clinical criteria for Parkinson's. And we know that both in Parkinson's disease that's established, as well as in the prodrome, so in someone with REM sleep behavior disorder, they have signs of neuroinflammation. And we see that all over the body. So if we look at the gut, we can see it in the stool and the microbiome. If we look at the blood, they've got increased inflammatory markers. If we look at the cerebrospinal fluid, there's increased inflammation. If we look in the brain, we can see activated immune cells. And we've seen it on post-mortem studies, but also in novel PET imaging, which I'm going to get back to. This trial was designed quite a while ago, and it took a while to get up and running, but essentially it's a double-blind, placebo-controlled trial between two sites, University of Macquarie in Sydney, Australia, and the University of Oxford in the UK. It's sponsored or funded by Syntara, the pharmaceutical company, as well as Parkinson's UK. And it looks at the reversibility of neuroinflammation in prodromal Parkinson's disease, so people with REM sleep behavior disorder. I think Livia also covered this. REM sleep behavior disorder is a parasomnia. It's a known sleep disorder. It's strongly linked to the prodrome of Parkinson's disease. It's prevalent in about 1% of the background population above 60 years of age, and it's more common in men. The conversion rate, if you have RBD, is about 6% per year to something like Parkinson's disease, and it's characterized by loss of muscle atonia during REM sleep. And this essentially means that a lot of you dream during your REM sleep phases, and if you have RBD and you're dreaming, you can act out your dreams. So you can talk, you can fight, and people with RBD typically have quite violent dreams. Okay, so the drug that we're using in this trial, so the population is people with RBD. The drug itself is called PXS4728A, a very cool name. And we know that this drug irreversibly inhibits amine oxidase copper-containing 3 and monoamine oxidase B. And these two play a role in reactive oxidative stress and neuroinflammation. So on the animal models, we know that it irreversibly binds these two things, which leads to reduced inflammation and oxidative stress. We also know that this drug is safe and well tolerated because it's been trialed in other diseases, including retinal and liver disease. The study objectives, as I've mentioned, is to measure the amount of reversibility of neuroinflammation after giving this drug in a prodromal population over 12 weeks, primarily by using a novel PET scan called a TSPO PET. We also want to look at the safety of this drug in the RBD population, and we want to see what happens when we reverse inflammation on the PET. How does that translate into practice? So we want to see the effect of the drug on clinical scores, digital assessments that you heard a lot about, but also on your blood results and your LP results. So the trial is designed for people with idiopathic or isolated REM sleep behavior disorder that's confirmed on polysomnography. They must be between 50 and 80 years old. And this is a placebo-controlled trial, so your chance of getting active drug is 3-to-1. The entire trial includes 5 clinic visits, 2 PET MRs, 1 or 2 LPs, and then some digital tasks that you do at home. So in the initial screening window, it's about 2 weeks at the moment. You do a clinical exam, you have a PET scan to make sure you have inflammation in your brain, and an LP. And then at baseline, you'll receive drug, and then we'll see you after 3 months for the follow-up clinical visit and the second PET MR. And then we wash out the drug for another 12 weeks, and we do the last clinic visit to compare if there were changes. Is it permanent, or do you revert back when we stop the meds? So an update. So we've started enrolling for this trial. At this moment, we've got 9 people enrolled into the Syntara trial. We have 5 more people booked up to November this year, and we need 6 more bookings to reach our target of 20 people by December 2025. Just a note, we are doing this trial in parallel to Discovery and PPMI, so it's not additional visits. We do everything at once to make it as time efficient as possible. And then just to give you an idea of the wealth of information that we get from participation in research, even though we only have 9 people enrolled, we already have clinical data of 28 clinic visits. We've got 30 new PET MR scans. We've got 112 vials of serum and plasma. We have 100 vials of cerebrospinal fluids. We've got 28 new OPDC smartphone recordings, and we've got 126 days of actigraphy recordings. So this is just every single visit really gives us a lot to work with. Then in terms of results, so everyone always asked when they take part in the trials, when will they know whether they're on active drug or placebo, when will they know if the trial was positive. And in general, this is true, but also for this study, we only start the analysis once the trial is completed. At the moment, that looks like June, July next year. And at that point, you will also find out if you had placebo or active drug. So June, July, Sydney and Oxford will split up the data and the samples, and then we'll start analysis, and there'll be a resultant publication which is signed off by the sponsor. So you'll know if the treatment was positive or negative. But we have a lot of data, and all of that data we will also put into the context of the wider OPDC discovery cohort. So we can use the PET MR scans from our RBD cohort in this trial, and we can combine it with the PD scans that we have from discovery, and we can look at neuroinflammation in a broader sense as well. So Open Day next year will be very interesting. And then just to show you how this novel PET MR looks, so you can see this is an axial slice, so straight through the brain on an MR. And I'm going to play on top of that the PET data. So you'll see this radioactive tracer flooding into the brain and then latching onto some of the inflammatory cells in certain areas. So you can see the inflammatory uptake in certain areas of the brain. Okay. the fun part. I want to give you some feedback from the participants because I think it's important for the researchers to see the faces behind all the data points and the samples and the digital tasks, but I also want you to see how it is to be part of a trial. So we are very lucky to be funded by Parkinson's UK, and they've done a lot of effort to also help us with a media release of our first patient, Francis, who is in the audience, was recruited, and we had some media coverage in some outlets and also a publication on their website. But I then went further and asked the participants to give me feedback on 3 questions. The first one was, how did you end up taking part in research? The second one was, what were your hopes and fears going into a trial? And the third one was, what did you wish you knew before, or which message do you have for people thinking about taking part in a drug trial? It was quite an open question, so you'll see some of the answers combine more than one. But I just want to read the first participant. He gave me written feedback. So he said, from time to time, over the course of about 10 years, I found myself waking up inside a disturbing dream. All tangled up in the duvet as I landed heavily on the bedroom floor. At first I thought these events were the result of a previous very stressful period in my life, a kind of PTSD. I only decided to consult a GP when the falls became more frequent and I was getting hurt. My GP referred me to the neurology sleep disorder unit for tests, and I was diagnosed with RBD. Like most people, including many medics, I've never had heard of RBD. But now my symptoms and diagnosis made complete sense. Just knowing what the problem was had come as a massive relief. Obviously, there's a downside to the diagnosis, but I've always preferred to be ahead of the game, not behind it. So the diagnosis gave me something very valuable. Time to adjust my life and start doing exercise, start doing a number of things I hope will slow down the long-term effects of RBD. A lot more physical and mental exercise are now my number 1 priority. So I think this shines a light on the fact that there is still lack of awareness, not only about Parkinson's disease, but also the prodrome and symptoms related to it. I'm now going to play a video.
So I was diagnosed in 2018 after my partner had complained of me beating her up in my sleep. It was initially sort of told to me at the point that it can lead to Parkinson's, and then I was invited onto the trial, and I thought, well, both it sort of keeps me informed of what's going on, and hopefully sort of helps with diagnosing what will happen in the future.
Just going to show you more. I can't unfortunately play all of them because of the time. We're all going over time. If you had a message for people who want to participate in research, what would that be? What did you learn, or what did you wish you knew before you started on this journey?
Well, the first thing is I was actually quite honoured to participate. I felt that it was a way of giving back. Of course, I had concerns that I was only able to consider it was considered because I had a problem, and I really didn't know the depth of that problem. And even today, I'm not sure that I still understand the depth of the problem, although I know I'm on a road, but I don't know where I am on that road. And I guess the whole purpose of this is try to establish what the factors are, the markers are for Parkinson's disease. But in that process, and perhaps I hadn't fully understood, but in that process I was asked to go through CAT scans and MRIs, and also to have a lung puncture. And again, in some ways this made me feel this is a very professional study. But it is true to say I was slightly anxious, although I have had a CAT and MRI in the past, but I hadn't had a lumbar puncture. But it was undertaken by one of the doctors who I found quite charming, and quite honestly, I hardly noticed that I'd had the lumbar puncture. So I know some people might be concerned, but I don't think there's any particular concern in that regard. And during the process of this, I have, I think, as I mentioned before, become slightly anxious because it's made me more, let's say, aware of the problem, and I've read about it and realized there are some side effects here which are not exactly pleasant things to have. But look, I'm into this study, I'm happy to be in, and I'm happy to continue. And if you're given the opportunity, I would heartily support that. I would suggest you have a go.
Okay, one last one.
Right, number 3, in the questionnaire, thing to answer. What would I say to someone who's considering going on the trial? Well, firstly, if you're in the realms of being spoken to about the trial itself, I'm guessing, and it is a guess, that you're there because you've probably been diagnosed with some neurological problem, possibly REM sleep disorder, RIBD, and therefore you're already in the mix, if you will, and the results of this trial and other trials like it is probably what's going to determine your future. Well, we hope it determines your future. So I would say this: considering the state of the NHS, it's not a political statement, it's just a fact of what it is. And an example of this is that my immediate neighbour, the house that lives next door to us, is an elderly gentleman in his early 80s, ex-rugby referee, used to referee, in fact, sent me off apparently. I didn't know this. I didn't recognise him. He certainly recognised me for playing when I was playing rugby when I was younger. He's a lovely chap. We've been to Twickenham a couple of times together. He's been diagnosed with Parkinson's, and he, apart from the diagnosis and a couple of visits to a consultant, he's sort of in the dark a little bit, really. He's not seeing or getting probably, I don't know, the help. He's okay, he's fit, his wife's lovely, and they're great, and they're getting on all right. But nevertheless, it's considering the interventions that I've had with Karolien and various doctors before her, and on the programs I'm on, the Discovery, I get some great treatment, access to the great minds that work in this field, and that alone, for me personally, is enough to make me think favourably in terms of whether to go on the trial or not. It's okay. It's okay. It's good. I think it's worthwhile. If you've got the time and the energy, get involved. And there's a community out there as well. I mean, I don't particularly get involved myself, but there's some great people out there that are helping other people out, and you get to learn about that as well. You get to learn a lot, actually. I think I said earlier on, I found it overwhelming at times, but it's good. And we need people to step up and have a go. And you're in charge. When you do it, you're in charge. You can dictate the terms by which you take part in the programme, and it's, have a go. It's well worth it. Thank you.
So I'm going to end it there. I hope that from this talk you also realize that you can take part, maybe not in this trial or a specific project, but find something that you feel passionate about and that you feel you can take part in, and please come approach us. We're all slightly nervous but very friendly people. I also want to say thanks to all the participants currently in the Syntara trial. It's an interesting process, and also to the entire team that's helping: Michelle, Johannes, Adriana, Jamil, Ludo, the team from Dendron, as well as the Cambridge colleagues doing the brain scans. Thank you.